set of papers today. This is the 13th year that we've had a Regional Science Consortium Symposium, Research Symposium, and that's been going very well, so we hope we can kick it off the same way today. You might have guessed that our first speaker was not able to join us this morning. This is why we've delayed our start today. We'll try to keep with the schedule as much as possible, which may mean some downtime, just waiting for the clock to tick by, if we have time. We'll try to keep our speakers on time. I'll be giving them flashcards saying they have 5 minutes or 2 minutes or stop. We're going to move on.
Good luck.
Yes, thank you. And I also wanted to say that I'm really pleased to be here. This is my 13th year of doing this, and I'm very happy to have been involved, and glad that Jeanette keeps me involved, even in retirement.
That's right.
Our first speaker today is Chelsea Gale, and she's going to be talking to us about an analysis of diet and habitat-related variation in body size and ringed sawbacks.
Hello. So, a little bit redundant, but my name's Chelsea Gale, and I worked with Ashley Gibson, right there, Will Selman, and Peter Lindemann, who I will probably keep referring to as Dr. Lindemann, because I feel weird calling him Peter. He's my professor. And we looked at an analysis on the diet and habitat-related variation in body size in ringed sawbacks, also known as Graptomys oculifera. So the first thing I'm going to talk about is the female trophic morphology in the genus Graptomys. So first, you can see here, these are megacephalic females, and one down here, these are pearl map turtles. Megacephalic females tend to eat mostly mollusks. Most of their diet consists of mollusks. Then the next one would be mesocephalic females. Mesocephalic females eat mostly like a moderate amount of mollusks because their heads are moderately wide. And then we have the species we're talking about today, the ringed sawback, which you can see here and on the ends here. These are microcephalic females, which are narrow-headed females whose diet consists only partially of mollusks. They don't really eat too many. So over here we have the female, that's who I was just talking about, and down here we have the male. Males mostly eat soft-bodied prey such as insect material and plant material. So the ringed sawbacks are part of the sawback clade, which is also with the yellow blotch sawback over here, Graptomys flavomaculata. You can see that they have yellow blotches all over their back. The ringed sawback, of course, has very vibrant orange rings all over it. And then the black-knobbed sawback, Graptomys nigronota. They also have rings, but you can see they're much less vibrant in color. And the knobbed part comes from the sawback being more knob-like than pointed like the other clade members. So now we're going to look at some general trends in the diets of the sawback clade. On the left, I have the diet study on black-knobbed sawbacks by Dr. Lindemann, and on the right, I have some unpublished data that's currently in review for a journal by Will Selman and Dr. Lindemann on the diet of Graptomys flavomaculata. And I boxed the index of relative important values, and I highlighted the IRI values of sponge. And you can see across the board for both species, males and females, that they are sponge specialists, meaning that most of their diet consists of sponge. If you start really looking more at the numbers, you can see that Graptomys nigronota also eat a lot of algae and insect material, and the Graptomys flavomaculata also eat a lot of sponge, algae, plant material, insect material, and males and females both eat mollusks, but females especially eat a lot of mollusks. And what was found in both of these species' diets was actually wood fragments when you look at their fecal samples. Wood fragments they're said to be in fecal samples because when turtles are eating sponge off of submerged dead wood, they are also eating wood because it just comes off along with the sponge. So typically when you see wood fragments in diet samples, it's a good indication that they are eating sponge. So there has been a previous diet study by Cochlear on the diet of graptomes. You can see on the left that most of their diet consists of insect material and plant material. He found that conclusion by looking at the digested stomach contents of 29 museum specimens. However, he did not mention sponge pretty much at all. It was not even brought up. However, you can see on the right here, 66% of his 29 samples had wood fragments, which, like I said, is a good indication of sponge. So that's where I come in. This is very interesting, and since Graptomes oculifera is a threatened species on the endangered species list, I thought that it was really important to give their diet another look at, because protecting the species could also mean protecting what it eats. So I wanted to see if Graptomes oculifera was like the other sawback clade members in that they are also sponge specialists. And I also wanted to differentiate between male and female diet because Cochlear did not do that. The other part of my study is the habitat-related variation in body size. So Amy Fehrenbach and her team looked at Graptomes sabinensis turtles over here. The black bars you can see are representations of sluggish rivers. So sluggish is low gradient, slow moving, just low and slow rivers, as I will continue to call them throughout the rest of my talk. And turtles found in those types of rivers have larger plastron lengths. And the plastron of a turtle is like the bottom of its shell, its belly, essentially. And that is when it's compared to fast, rapidly moving sandbar-dominated areas, which are represented by the white bars. You can see over here that Graptomes flavimaculata and Graptomes nigronota both also show this trend of being larger in sluggish, low and slow rivers. However, this hasn't really been looked at much for Graptomes oculifera, so I wanted to take a look at that because I hypothesize that they will also, like the other clade members again, show this trend in being larger in sluggish, low and slow rivers. So our study area, Graptomes oculifera is endemic to the Pearl River system in Mississippi and Louisiana. So the orange dots are representations of the fast, rapidly moving sandbar-dominated areas. And there's a couple of those, but what's really important that we wanted to look at are these white areas. And these are the areas where we expect to find the larger sized turtles, because they're the low and slow areas of the rivers. Down here, we have a coastal area. This is the East Pearl River. The site we went to was Napoleon. And it's a coastal area. It's actually it's less than 2 feet above sea level, so it really is on the coast. And then up here we have Gillahachie Creek. You can see it better there. Gillahachie Creek is a low and slow river because it is a creek leading into a reservoir. So for our field methods, we caught turtles using mostly open-top basking traps, which, look at this picture, isn't that a good picture? Look at that turtle. This is an open-top basking trap. And what we would do is we would see turtles basking and we'd be like, oh, that looks like a great spot to put a trap. So we would go over, the turtle would swim away, we tie the trap under the tree, leave, go put more traps up, and then when we came back, the turtle would be back, we'd approach it, and then boom, just like this picture, we'd jump right into our trap. And then these are fight nets, which is open on one side, so the turtles would just swim right in there, and you can see a float here, so the turtles were able to go up and get air. After capturing the turtles, we use a flexible ruler to measure their plastron length, which, like I said, is the underneath their shell right here. And then after that, we would use calipers to measure the carapace length and height with calipers, and then we use spring scales to get their mass. And if growth lines were visible on the plastron, because with age their plastron kind of smooths out, then it can be difficult to see their age. But if they were visible, then their age was noted. So the difference between males and females. So you can see on the left pictures, these two here, they're actually the same turtles in both pictures. Females are much, much, much larger than males are. And in fact, in this picture, the female's plastron length is actually 160 millimeters, and the male's only 83 millimeters. And according to a study by Jones and Hartfield in 1995, the male here is actually closer to being full grown than she is. So, crazy. It is a little more difficult to tell the difference in size between juvenile females and adult males because they're about the same size. So in order to tell the difference between them, we looked at their tails. Males have very, very long tails with the cloaca reaching out past the rim of the carapace. And females, the cloaca about matches up with the edge of it. Males also have these very long claws, which I learned the hard way will cut your skin open. So marking the turtles. Turtles were marked using a file. They were given a unique number system, like you can see here, 4-4 and 4-10, in which we would file on their marginal scoots, which means that were scoots here. Marking the turtles helped us to keep from any resampling. So you can see here, this is 4-4, so you can go 1, 2, 3, 4. You can see a notch here, and then 1, 2, 3, 4, another notch. And this is 4-10 here. So collecting the samples, we would keep turtles in plastic buckets filled with water for 1 to 2 nights, depending on how long it took to get a good amount of fecal samples. And then in the morning, we would wake up and we would use a sieve here to strain the water out and put the fecal samples into labeled jars with 4-4 and 11-1 here, their marginal scoop numbers. And those were filled with 70% ethanol. And after that, we would release the turtles, which is always the best part of the trip. So for the data analysis, we used a stereoscope to identify prey items and categorize them into, like I said before, insect material, and then you have like a million different types of insects that they could be eating. And the volumetric displacement was used with a graduated cylinder to record the volume for each sample. And then up here, this is the equation for the index of relative importance, which was used to compare the amounts of prey that was each prey category eaten by males and females. I represents the prey category, VI is the mean total volume of the category, and FI is the frequency of occurrence of each category. And I have some examples down here. This is dark false mussel over here, what it would look like under a stereoscope. And this is actually some filamentous algae up here. The dark false mussels are coastal species, so we would really only expect to find these in Napoleon because, like I said, it's a very coastal area. This is what sponge looks like. This is the main thing that we were trying to find. And with sponge comes spicules and gemmules. All these little white ticks covering the whole entire bottom of this watch glass here. And these gemmules, 1, 2, 3, 4 of them here. Spicules and gemmules actually resist the digestive process. So they're very good identifying features of sponge being in fecal samples when it's not as clear and visible as this here. So these are the results from my study. Again, I boxed the index of relative importance values and highlighted the sponges. And you can see here for adult males and adult females, they have the highest IRI value being for sponges and for juvenile females, it is the second largest IRI value. So this is very good support for my hypothesis that, like the other clade members, Graptomys oculifera are sponge specialists. Insect material and plant material really kind of make up the rest of it. And you can also see over here the 7 and the 5 compared to the 1.3. Females, again, eat a lot more mollusks than males do, so we expected to find that as well. So, makes sense. And I also just want to point out that we had a total of 34 samples, and in all 34 samples, sponge was found in every single one, and wood fragments were found in 32 out of the 34 samples. And the 2 that they were not found in also happen to have the lowest trace of sponge. So for habitat-related variation in body size, well, we can just look at the female and male at the same time because they had the same results. As I said before, Napoleon and Killahatchee are the low and slow areas where we expect to find the larger sized turtles, the longer plastron length. And those are indicated by the black bars. And you can clearly see in my boxes, that's exactly what we found. When compared to upstream, fast-flowing areas, Graptomys oculifera were bigger in the downstream areas. And this could potentially be due to thermal stability, but also due to the fact that low and slow areas have better productivity because the slower flow of the water, it allows nutrients to remain in one area longer, and organisms will take advantage of that and use those nutrients for growth. So I just want to give a quick acknowledgement to the U.S. Fish and Wildlife Service, who has been providing funding for this research project for the last 30 years. Much appreciated. And that's all I have for you today.
Do these turtles have any particular adaptations for eating sponges? Anything?
Not that I'm aware of. Do you know, Dr. Lindemann?
I guess to be determined whether there's anything about the digestive tract lining that allows them to process these spicules.
You know, a mouth is made for like picking up sponge more so than another turtle.
It's very, besides the very narrow edge, it's very narrow alveolar surface on the inside that may help them scrape things off a little bit.
Good idea for a future study, I say.
Is there any seasonality to the availability of the sponges? As you showed that earlier study that didn't really have any sponges in the diet, although it had the wood fragments. Is it something that maybe the sampling times were different?
I don't think it's seasonal at all. Cochran did not find any sponge. We speculate it's just because he wasn't able to recognize it in digestive contents. It's a lot more visible in fecal samples, which is why we chose this method. Any other questions?
Is sponge high nutrition or low nutrition food? Kind of a spicule and stuff, or much substance to them?
I would guess the spicules aren't providing anything, but the soft body stuff that is more digestible. Where that would rate, I don't even know if anybody's looked at that because there are actually very few animals that eat sponges and specialize on them. So that's a question.
Good questions, because not many of us know the answer. Anything else?
Thank you.
Thank you.
Our next speaker is Ashley Gibson, and she's going to be talking about the evaluation of range and population estimates of two endangered species, candidate map turtles. Which one do I want to have here?
I do, yes. Thank you. Okay. Good morning, everyone. Thank you for joining me today, and thank you for that introduction. Today, I'll be discussing two species of Graptomys turtles and their current ranges and populations. Graptomys is a genus of freshwater aquatic turtles found primarily in medium and large rivers. They are commonly referred to as map turtles and sometimes sawbacks, which describes the characteristic ridge along the midline of the carapace. You can see right here. This past summer, I had the pleasure of assisting Dr. Peter Lindemann with his research on Graptomys pearlensis and Graptomys gibbonsi. They are commonly referred to as the pearl map turtle and the Pascagoula map turtle, and they are named for the rivers they each occupy in southern Mississippi and the southeastern corner of Louisiana. Our understanding of their range and habitat has evolved over the past several years, with the most up-to-date range covering most of the two river systems. In 1893, as you can see, very little was known about them. The researcher here used museum specimens to develop this range. Between 1893 and 1958, virtually no research was done on these species, and it wasn't until a Tulane professor and his students started collecting specimens in the rivers that they developed a much better understanding of the range. But even then, until 1971, the University of Southern Mississippi developed a lot more in the Pascagoula River, going into the tributaries. Dr. Lindemann came in 1999 and added to the Pearl tributary. And in the early 2000s, Will Selman from Millsap College, he worked a lot on the Pascagoula River system. He did a lot of work in these tributaries, adding to our knowledge. He did a little in the Pearl as well. Finally, we see here, this is the current range we have. And compared to 1893, it's a lot better. They have a very wide range. So both of these species coexist with another Graptomys species. The pearl map turtle lives sympatrically with the ringed sawback, Graptomys oculifera, and the Pascagoula map turtle is sympatric with the yellow-blotched sawback, Graptomys flavimaculata. Both of these congeneric species are already listed on the endangered species list as threatened, one in 1986 and the other in 1990. So why haven't the pearl and Pascagoula turtles been listed yet? Well, in 1893, the pearl and Pascagoula turtles were listed as one species with the Alabama map turtle, Graptomys pulchra, together with the Mobile Bay and Escambia Bay populations to the east in Alabama and the Florida Panhandle. A division wasn't made until 1992, but then the pearl and Pascagoula turtles were listed under one name as Graptomys gibbonsi. It wasn't until 2010 that the map turtles in the Pearl River drainages were determined to be genetically and morphologically different from the ones in the Pascagoula River system, finally leading to the description of a new species, Graptomys pearlensis. Now, it's easy to understand why these species were considered one for so long. They're very similar, and unless you know what you're looking for, it's very hard to tell them apart. However, because of this, these species were the subject of little research, and we are only now finally starting to understand their ecology. They are now under review by the U.S. Fish and Wildlife Service to be placed on the endangered species list. It is believed that their population has been declining over the years. However, due to the taxonomic changes, any research done on them was secondary, or the data was collected when they were still considered one species, Graptomys gibbonsi. Still, if we compare the ratios between them and their congeners, we're finding it was once approximately a 1-to-1 ratio, is now more of a 5-to-1 ratio. That's 1 Graftomys gibbonsi, or Graftomys perlensis, per congeneric sawback. And this was based on a study done in 1994 to 1995 by Dr. Peter Lindemann. In order to establish boundaries on their territories, bridge counts and boat cruises were conducted along the lengths of both of the rivers, including their tributaries. Map turtles are habitual basking turtles, often found lounging on dead wood on a warm, sunny day. We used image-stabilizing 18x binoculars to locate and identify these turtles to species. The binoculars gave us a vision range of approximately 150 meters. The surveys were restricted to the summer months from May to August and were conducted on days with little to no rain to ensure high basking density. The data collected were species, county, tributary, and number of turtles sighted. In addition to surveys, we also captured turtles using basking traps and fyke nets. Basking traps are cylindrical traps that we put out daily and collected every night. They are secured under a piece of wood, of dead wood, or a branch sticking out, something we think a turtle might be sitting on. And then the turtles fall in when they jump off the wood. Fyke nets are a little more complicated. They're a lot heavier and messy. You have to secure both ends to a secure object, like a tree, and then they're partially submerged in the water. The one downfall to these traps is that sometimes you catch more than turtles. Actually, a lot of time you catch more than turtles. Like, we caught an alligator this summer, and I have to say, that was the highlight of the trip. That was a lot of fun. So although the trapping wasn't empirical to our calculations, tracking how many pearls and passagulas we caught was another way to record the abundance of each species. Currently, the study has been ongoing for 3 years now, from 2015 to 2017. We also incorporated earlier data from Will Selman's 2006 to 2008 data collection that he did for his PhD project. And data was also collected sporadically between the years of 2007 and 2013 by Dr. Lindemann. For each tributary, we calculate a basking density of turtles seen in point counts from bridges and other river access points, and turtles seen per kilometer during john boat or canoe cruises. The data were then organized from high basking density to very low density and assigned a characterized basking density for each category. For larger tributaries that showed uneven distribution, we split them into upper and lower halves and then assign them to each of the categories of high, very low density in between. We then took the characterized basking density of each category and multiplied it by the tributary's length in kilometers. Based on previous studies that use RegSciTV techniques on Graftomys species to estimate population size, we assume that the basking turtles we saw constituted only 20% of the total population and multiplied everything by 5 in order to get a final population estimate. The current tracings on this map are the most upstream and downstream known extents of these species. So the total lengths we currently know of their ranges are approximately 1,300 river kilometers for the pearl and a little over 1,700 river kilometers for the Pascagoula. The difference between them is that most of the length in the pearl is in its main stem, which goes all the way here, and for the Pascagoula, it's mostly in its tributaries. We found that in the Pearl River, the majority of the turtle population was spread throughout the main stem. And the Pascagoula had similar findings with 72% of its population in the main stem, including the Chickasaw and the Leaf River. Our final totals are as follows. There is an estimated population of almost 19,000 pearls and 30,000 Pascagoula turtles occupying these river systems. There are 58% more Pascagoulas than there are pearls. Besides the main stem, about 35% of the pearls are found in the major tributaries, which include Labucha, Yukonukane, Pelahatchie, McGee's Creek, and the Strong River and the Boca Chica. Only 1.6% were found in the minor tributaries. For the Pascagoula, outside of the leaf and Chickasaw in the main stem, so outside of these two, well, these three, there was very little population of turtles. Only 22.3% are found in the major tributaries, which are including Okeechobee, Tullahoma, Bacatuna, Suwanlovey, and Chunky Creek. There is a mere 5% in the other minor tributaries. Oh, here's the estimated populations. In addition to a higher estimated population, there were more Pascagoulas caught per trap day versus pearls. A trap day is when we leave basking traps out daily, we leave basking traps out and then we check them hourly, or when we leave fyke nets out overnight. Out of 54 pearls caught, it averaged 1 pearl for every 7.7 trap days, and for the 97 Pascagoulas caught, it was 1 for every 5.8 trap days. This shows a higher density of Pascagoula turtles. Based on these preliminary numbers, we are suggesting a status of threatened for the pearl map turtles and threatened by similarity of appearance, abbreviated TSA, for the Pascagoula turtles. TSA is a special category on the endangered species list, and it's exactly what it sounds like. The Pascagoula turtles are surviving much better than the pearls. However, due to similarity of appearance to the pearls, anybody could pass off a pearl as a Pascagoula on the pet trade and vice versa. If both the species are protected, it will simplify the process and make it easier on authorities. Having the Pascagoula turtles listed as TSA doesn't require a recovery plan. However, the pearls will need one. Due to the extensive branching on the Pascagoula, if a reservoir or other major change were made to the river, it wouldn't affect the rest of the river system, and the Pascagoulas should be able to survive. However, if the pearl is very linear with very little branching, if a reservoir or any other major change were made to this river, the pearls, it could be detrimental to their population. There's the other option of listing Pascagoula map turtles as threatened as well, because their congeners species greatly outnumbers them still, and they could use some help to even the odds. Basking surveys will continue for another year before final estimates and recommendations are made. I would also like to thank the U.S. Fish and Wildlife Service for funding this research, and a thank you to Dr. Lindemann for providing me with such an amazing opportunity. Thank you, and does anybody have any questions?
Questions for Ashley?
None.
How many different species of map turtles are in these river systems?
In these river systems, you have 2 in each one, the pearl and the ring sawback that Chelsea was talking about in the pearl, and then you have the Pascagoula and the yellow blotched in the Pascagoula River.
Those are the only ones that occur in those rivers?
Yes.
Can you tell the two species apart when they're very young or in a juvenile stage, or do you have to wait till they're adults to distinguish the two species? Or did you have that issue at all when you were sampling?
Well, when we were sampling, since we were in different rivers, it was like, oh, well, we're in the Pascagoula, so it's the Pascagoula. That made it easy. There are some telltale signs, like the dark ridge on their, they have a dark line that goes down the center of their carapace. In the Pascagoula, I do believe it's a broken line. In the pearl, it's a solid line. However, it's not a very reliable way sometimes, so even when they're juvenile versus adult, it's still a little hard.
Were there times when you said, we're not sure?
There were times when we messed up, like, oh, that's a pearl. But like I said, if we were in the Pascagoula River, we knew it was a Pascagoula, and if we were in the Pearl, we're like, that's okay, that's a pearl. So.
Was the question about telling the sawbacks apart from the pearl and Pascagoulas within each drainage? Or was it telling the difference between the drainages?
Telling the difference between the two species in the same drainage.
In the same drainage?
Oh, in the same drainage. Oh, so between the sawback and the... Oh, they're very different. The sawback has the rings on it. They were what was in Chelsea's show. They have a very small head, and the pearls have a more pointed head, very massive head when they're large, and they honestly just look completely different. So it's crazy. Yeah. I'm sorry for misunderstanding. Yes.
Do you know what some of the main reasons are for the species to be placed on the endangered species list?
Well, their populations are declining.
Is there a reason for that? Is it hunting or loss of habitat?
I'd have to say loss of habitat, and there's the pet trade. They're a very unique turtle. People want them as a pet. Habitat changes are trying to be made to these rivers. Like there's a suggestion for a reservoir to be built in, or even a boat canal to be dug out for, was it the Pearl River? Which, as I said, is something like that would affect the entire Pearl River. And since there is no drainages for them to escape into, that would affect their population. So.
Okay, thank you. Our next speaker is Joe Seacrest from the Purple Martin Association, and Conservation Association, pardon me. He said that he might go a little bit long, so we're going to go ahead and start a little bit early. So maybe come back into sync again a little bit.
All right, I'll do my best to get us back on time here. All right. I can't use this little bitty red laser. When you're talking purple martins, you have to use a purple laser. Okay, so I want to talk a little bit today about some of the research that PMCA and our partners have been doing over the last 10 or so years. So when I say we, I've only been here for the last few years, so sometimes it'll be the royal we, sometimes it'll be we, me, and the partners. So. Purple martins are North America's largest species of swallow. They're named purple martins because of the color on the adult males right there on the left. It's kind of an iridescent blue, really, but they've got the name purple martin, and that's stuck. This is a 1-year-old male, but it looks very similar to what females look like. So the males are where they get their name. It's a species that has 3 subspecies. Primarily, it consists of this eastern subspecies here, Prognes subus subus. That's like 98.5% of the total population. There's a couple other subspecies up here that don't have quite the same conservation issues that the eastern subspecies has. So that, you know, with the population focus being on the eastern subspecies and most of the conservation issues, that is where the focus of our research lies. It's a aerial insectivore, meaning it eats only flying insects, and an estimate is that they're responsible for eating 262 billion with a bee insects every year. So this is an important part of our ecosystem. They're doing quite an ecosystem service to us, so they're an important bird to conserve. Part of the interest with purple martin lies with the long relationship that they have with humans. Pre-European settlement, before we really have well-recorded history, Native Americans began hanging hollowed out gourds, and purple martins began nesting in them. And for years and years and years, the purple martins would nest in these artificial cavities. They'd nest in natural cavities, being woodpecker holes in trees. They're a secondary cavity nester. They'd also nest in, holes in rocky cliff faces. They thrived nesting in both places. We don't really know why Native Americans began doing this, whether it was for insect control, the obvious first guess. They could also have acted as a watchdog. They do alarm calls when they don't recognize somebody coming by. They could be scarecrows. They could have chased crows or other nuisance birds away from the crops. They could have been just like a calendar. They show up like clockwork from their migration every year. I know that here in Erie, they're going to show up first week of April every year like clockwork from their migration. So those are some of the guesses that we have on why it may have been the case. And European settlers came along and they thought, hey, this is a pretty cool thing that these birds show up and they will nest right alongside humans. This is around the turn of the 19th century. You can see a martin house up there with a little ladder so they could climb up and check it. This is in Nebraska, I think. But purple martin houses speckled all over, spread all over North America. Obviously, the settlers went with a little more anthropocentric housing type, the typical birdhouse that you'd think of. And then, as is the case a lot of times, humans screwed things up. 1850, around 1850, European settlers decided they wanted a little taste of home, started releasing English house sparrows in an effort to, get this great bird that they missed so much established in the United States. And get established it did, began to spread. And then in 1890, a group in New York City that really loved William Shakespeare. They wanted to release all of the birds that are mentioned in the works of William Shakespeare. And they released them in Central Park, and everybody was thrilled, applause. And that bird immediately began to spread as well, and along with the English house sparrow, spread across North America. And the problem with purple martins, or for purple martins, is that these birds are also secondary cavity nesters, and they will outcompete purple martins for those natural cavities. So we've got the obvious loss of habitat with deforestation as settlers moved west. You think now if you see a dead tree, what do? You cut it down, right? You don't want a dead tree around. So that limits the number of natural cavities out there. And then for those natural cavities that do remain, we've got these two invasive species that are outcompeting purple martins. So the end result is that eastern subspecies that makes up the majority of that range map, they rely completely on human-provided housing for nesting. They don't nest anywhere naturally anymore. No more natural cavities in the eastern United States at all. So you can see, there's different types of housing. There's gourds that simulate what the Native Americans would provide, but there's also wooden houses and metal houses. You can see there's one out in the parking lot as you come in. We have colonies on the park as well. One of the pros with this is that close relationship with humans allows close monitoring of this bird that is now relying on us. And the PMCA has worked for 30 years to establish best management practices, to monitor the species. So you can lower the housing down. The birds, half the time, they'll stay in the nest. You could take out the adults, hold them in your hand. They're practically, at times, a tame animal. They are still quite wild, though, and you'll see from the migration that they spend the majority of their time elsewhere. Here's a shot there of a little 4-day-old nestling getting aged at one of our study sites. So despite all of our work to maximize the production of this species, its populations are declining in many areas. This is from the Breeding Bird Survey. Those red areas that dominate the northern part of the range with some areas of decline down in the southern part of the range, those in the last, I think this is from 1966 on, those have had an annual decrease of greater than 1.5% each year. So that's populations that are tanking right there. And range-wide, since 1966, there's been an overall loss of 0.87% of the purple martin population per year, and that's equivalent, when you calculate compounding interest on the population, of 1/3 of all purple martins across the range lost in that time in the last 50 years. Here in Pennsylvania, it's actually quite a bit worse. We're looking at the loss of about 3/4 of purple martins in that time. But like I said, we have this migratory species. We're doing what we can here in North America, but the population decline remains, and there are species that spends the majority of the year away from us, away from the breeding ground, down in the tropics, the Amazon rainforest in South America. We lack a thorough understanding of what happens to the purple martins away from the breeding grounds down there. So just a background on some of the research and how difficult it's been to really even establish bird migration. I thought this was cool. Some early beliefs on bird migration. They knew that the birds disappeared. This guy, Swedish Archbishop Olaus Magnus, in a book, he explained the thoughts that not purple martins, but swallows in Europe, which are purple martins and swallow, each year as winter came, they would sink to the bottom of ponds and lakes and spend the winter down underwater. And this is actually an etching from a book of fishermen, and those aren't fish in there. There's wings on those fish. Those are birds. They're pulling up a net full of swallows. And then Aristotle, he actually had maybe a better explanation. He thought that the species just changed from one to the other. So this is a common European redstart, and he thought that as winter came, it converted to the European robin, because this species migrated south. He didn't realize this one came from north down. So they're kind of a similar species. Maybe he was a little more onto something. Smart guy, that Aristotle. I guess that's why we remember his name. But then along came bird banding. The first kind of established in an organized fashion here in the United States in 1902. PMCA bans a lot of birds every year. We've done about over 20,000 birds that we've banded. And that just is putting numbered or color-coded leg bands on the birds so you can see where they go and learn a lot about the species once you know what an individual is doing. So since the organized bird banding started in 1902, there's been over 500,000 purple martins banded across North America. And anybody want to take a guess on how many times those birds have been encountered outside of the USA and Canada? It's not many. 39. 500,000 birds got banded, 39 spotted outside of North America. So we do have direct evidence that, hey, these birds are the ones that are going down south. So we've established that much of migration. We don't know what happens along the way. And really the majority of these encounters are when they find a dead purple martin. So we know that's where they died. We don't know what they did along the way, what they were doing down there. Maybe this is where they're found is where they don't want to be because that's where they died. So, you know, that's a piece of the puzzle that's established through bird banding. It's not really the primary, you know, objective of bird banding. You can learn a lot more things from it other than migration, but it's a piece of the puzzle. There you can see lines connecting where they were banded and where those were recovered. Kind of a mishmash. You know, there's something to be learned there, but it doesn't really paint a great picture. The curiosity also extended down to the Brazilian side of things with a scientist down there, and you'll see from this clip from a Brazilian documentary what they did.
O engenheiro químico e apaixonado por passarinhos, tal das fish, ainda era menino quando abateu uma andorinha azul para o trabalho de pintura do parque. Nos anos 80, com a ajuda dos pesquisadores Dino Visor e Jacques Villiard, constatou que era mesmo a Progne subs, a andorinha azul que vinha da América do Norte. Faltava confirmar a migração e a rota. Através da Associação de Preservação da Vida Selvagem, Dalgas conseguiu uma autorização do governo americano para a microetiquetação. Um tinte especial pintou mais de 300.000 andorinhas em 5 cidades paulistas. Cada grupo recebeu uma cor diferente, só visível com luz ultravioleta. O trabalho em conjunto com sociedades andorinheiras do Canadá e Estados Unidos foi um sucesso.
Com a pintura das andorinhas, 2 meses depois, foram recuperados nos Estados Unidos e Canadá 32 aves, o que provou definitivamente que elas migram. Agora, com a recuperação das penas que elas perderam nos dormitórios durante a volta, foi possível descrever com exatidão a rota migratória deles.
So you see there, I just think that's so cool that they were able to do that. That was in the mid-'80s that they painted all those hundreds of thousands of birds down in roosts in the Sao Paulo area. And then they had coordinated folks up here in North America that would pick up feathers that had been shed or lost and checking them under blacklight to see which ones match up. They actually used different colors for different roosts down there, so they even took it a step farther. So, we're kind of drawing a picture of migration, but we didn't really start to learn a lot of it until about 10 years ago. This device was developed called a light level geolocator. And it's an ingenious little device that basically it just looks at light levels. And you can tell where something is in the world by light levels. You can tell how far east or west something is by the time of sunrise and sunset, just like with time zones. And you can tell how far north and south something is by the length of the day, just because of the tilt of the Earth's axis. So from that, we can determine, with some error, obviously, where an animal is. So it got miniaturized enough, and battery technology got miniaturized enough for this to be able to put onto migratory birds, purple martins. So we deployed these in locations all across North America. Initially, this research was started by Bridget Stutchbury at York University in Ontario, and has since been taken up by Dr. Kevin Fraser at University of Manitoba, along with, obviously, us, the Purple Martin Conservation Association, and another major partner being Disney's Animal Kingdom down in Florida. But we also had these going on with a lot of other groups that were working with us on this research. And we're able to tell stories like this. This bird is a 1-year-old female. You can see her little light level geolocator sticking out there. And she got her device put on in Alberta, Canada. right after breeding season. So you can start to see things like this. You see that in 2 weeks after departing on migration, she ended up down Louisiana, ready to jump across the Gulf. And that she did. And then she spent about a month relaxing in the Yucatan, refueling, plenty of insects to eat in the jungles of the Yucatan. And this is pretty typical of what we see of the migration paths of these birds. Made it down to the Amazon rainforest, and that's about, what, 2 months? Finally reached down to the final destination and spent the winter down there. And then departed on April 24th like a shot. Made it up in 1 week all the way back to the Yucatan, and then within 2 more weeks it was all the way back. So 3 weeks it made that full trip all the way back. So the time away from the breeding habitat was 263 days, 22,000 kilometers for the round trip, and that spring migration was 21 days, giving it a speed of over 600 kilometers per day on that return trip. So they're really hustling to get back. And you can see here a composite of some of the migration tracks from here at our study site here in Erie. And you can start to draw pictures. You can see that, hey, there's areas of importance where these birds converge along the way. You know, we should look at conservation issues and the habitat use in those areas. And we're able to put together cool animations like this. I love this. This is an animation of several years of purple martins from here in their migration. And you could just sit and watch this forever, because each one of them is doing something cool. I like this little last guy there. He hangs around and he's like, oh, I better get going. So, you know, we're starting to paint a picture of migration. And we're able to look at stopovers because they're taking these breaks at places. And you can see the importance of certain areas. You can see here how important the Yucatan is. The size of these circles is the length of the stopover. So you've got plenty of birds hanging out for a month or more. You know, all the rest, you know, they spend some time in places, but they're kind of transient. But you can start to see importance of areas. And we thought, you know, let's look for the first time at migratory connectivity. And that is a term that means birds that live together, you know, in their breeding season. Do they stick together down in their overwintering season? Do they all kind of have their own territories down there as well? And we found that, at least with that eastern subspecies, they're all kind of the same place. Everybody's just hanging out in the Amazon, going all over the place. Interestingly, though, but it's with a small sample size that we're actually looking to expand on, that northwest subspecies, it was the only one that was showing up down in that Sao Paulo area where that little clip of the documentary was from. So very loose migratory connectivity. And then, you know, in that we're looking to see, you know, if you think back about that map where it had the red areas that were decreasing population, you know, maybe those birds from those areas are going, and where they're going down in Brazil are areas that are in trouble. You know, they're losing habitat, they're being, you know, they're not having enough food, what have you. But we found that the ones from those northern breeding populations that are not doing well, their habitat use was basically the same as the central and southern populations that were, you know, their populations were doing fine. So it doesn't seem to be, at least at a broad scale, what is driving the population decline. And then technology marched on, and we got GPS technology shrunk down enough to fit on these birds. So this is a GPS data logger, and that's taken our accuracy from 200 kilometers, which is what the accuracy of those geolocators are. So draw a big circle on the map, you know the bird's somewhere in there. You don't really know exactly what habitat is being used, to less than 10 meters. So you can go from knowing a bird is in the Yucatan somewhere to knowing that it's in this tree in the Yucatan. And our first year of data was pretty cool. So each of these colors is a different bird from a different place. And you know, it's pretty typical of what it looked like there, but we do have these areas, especially that one right there with that little pinwheel. And in that pinwheel, that spot, we put on GPS tags at 6 different sites. We had 5 different sites, birds from 5 different places, going to the exact same little sandbar and a tributary of the Amazon. So we're finally able to get from broad scale habitat use to fine scale, and these islands start popping out at us. What the heck, you know? Those birds are from all over the place. All of our other research shows that the birds are going all over. You know, there's no real rhyme or reason to the habitat uses. They seem to be using all of the Amazon. They're just eating bugs all over the place. But these little places, this is birds from 2 different places. I think that's Florida and I want to say Minnesota. Maybe that one's Florida, Minnesota. But you know, really graphically disparate colonies that these birds came from. We're like, we got to go see what's going on. So we picked this little area called the Anavillanas. It is a archipelago of islands in the Rio Negro, which is, these are the 2 tributaries that make up the Amazon River. This Rio Negro, this is the Rio Solimões. Down here would be the Amazon. So we hopped on a boat. We went up tributary of the Amazon to see, you know, what's so important. We had to be on the ground to see what's going on with these islands. So here we are. We're cruising up the Amazon. And we made landfall. This is us setting foot on one of these islands that the previous year had been visited by purple martins. And we find that it's these sandbars really devoid of ground vegetation. And it has kind of unique river-adapted tree species. But one of the things we didn't realize, here's another shot of it, kind of from down the, and there's our boat in the background. The Mamie Denise was our boat. So this is the same island we were standing on there. This is kind of a fuzzy shot, but it's from space. So I have to, you know, give NASA a little bit of leeway on focusing their shots. But this is during the dry season, and that's when we were there. And one of the things we didn't really realize until we were there and experiencing it, that the river levels vary up to 15 meters. So we're talking 45 feet between a wet season and a dry season. And we were there at the driest of the dry season, but this shot is from the wet season. Just the treetops is all that's left. And that's pretty typical of these islands that it seems like the birds were using. And our best guess is that, you know, and this is ongoing research, that was just the first year, is that these are completely scrubbed of ground predators every year. So this, and the width of the Amazon, at least in places, is 15, 20 kilometers. It's like a lake when you're sitting there on it. So these are relatively protected areas where the birds are safe and they're able to spend the night, because that's when we were getting these GPS fixes, was at night, because during the day they're flying all over the place. And unfortunately, of that first year's worth of GPS data, only 17% of those overnight roosts are in protected areas. So what the PMCA has been doing, we've been working with our partners that we found down there to help to protect those areas of importance, the habitat that seems so important to these birds, because they're greatly vulnerable to, you know, development. Anything that would change the water levels obviously would greatly affect this habitat. So thanks all for coming out, and any questions?
One question for Joe. When you only have one, it's got to be a good one.
I have a question.
Yeah.
Are there invasive species down in South America that take over places for the purple martin and nests as there are in North America?
Yeah, it's a good question. They only nest up here in North America. So down there, they're not using any nesting sites. They're just hanging out. They're spending the nights roosting in trees or on man-made structures, which is the case a fair amount of the time. But they're not running into direct competition like they would be up here from invasive species. Yeah?
Now, your data, and apologize if it's wrong, you were showing that the birds all over the U.S. tend to go in the same areas down in the Amazon, and only the northern populations in the U.S. are being really hard hit in population declines. So does that not then indicate that we have issues up here?
Absolutely, absolutely. Yeah, more and more we're realizing that a lot of the causes, are up here, and it would stand to reason, right? With the species that's completely reliant on humans for nesting habitat, obviously things are not great up here, but drawing an entire picture of the life cycle of the bird, helps, because every little bit that we can do to help this species to continue to exist. So if there's a bit of habitat down south that is just critical and we can get that conserved, just a step in the right direction.
Are there fewer people putting out purple mountain cardinals?
Absolutely, absolutely. Yeah, it is a hobby that is primarily something that's done by older folks, and it is becoming less popular, and that is a major part of our mission at the PMCA, besides doing research, but doing education to help folks know that this is a species that needs us. It needs more people. As long as there are people putting these housing up, those birds will continue to exist. Every purple martin in the sky east of the Rockies came from somebody's backyard.
Thank you. Our next speakers are from Genus University, and I'm not sure if we have two speakers or one. Two. They are Maria Colt and Ahmed Kuliev, and I'd like them to come up and tell us about the bat population in Erie.
Thank you, Dr. Covert. Thanks everybody who was able to make it today. My name is Maria Colt.
And I'm Ahmed Kuliev.
And today we're going to be presenting on the bat population on Genus University's campus in Erie, Pennsylvania. Okay, to start off, I'm just going to talk a little bit about bats in general. Bats are the only mammals that are capable of actually flying. There's over 1,100 different species of bats, making up close to a quarter of all mammal species. They can live pretty much anywhere in the world except for extreme deserts and polar regions, and that's because there aren't any insects there for them to eat. They are nocturnal, so they sleep during the day and they eat at night. And they have many important ecological factors for us. They pollinate flowers in South America, they disperse seeds, and they can eat millions of insects. And bat numbers are decreasing due to the presence of white nose syndrome, and that's in hibernating bats. So white nose syndrome is caused by the fungus Pseudogymnoascus destructans, and it grows optimally in caves because of the constant temperature and the constant humidity that they provide. Over half of our U.S. bat species are cave hibernators, so potentially those half could be affected by white nose syndrome. And the fungus, when it gets on the bats, it gets on their face, their ears, their tails, or their wings. Here's some pictures of bats with white nose syndrome. The bottom left, you can see it would be especially detrimental if they get it on their wings. Because they're so thin, they could be perforated easily, and then they would have a harder time flying and could potentially lead to mortality. So what pretty much happens when the bats get white nose syndrome? They'll go to hibernate, wake up out of hibernation, like itching themselves, wondering what's on them. and then they'll use up a lot of their fat reserves. So they leave the cave looking for insects to get some energy back, and they can either die due to starvation or the cold temperatures. Scientists have seen mortality of 100% in some caves, and since 2006, when white nose syndrome was first discovered in the United States in around Albany, New York, 6 million bats have died. And some researchers claim that it's the most precipitous wildlife decline in the past century of North America. So you can think of this similarly to the bison die-off in the late 1800s. Here's a map of the current white nose syndrome status. You can see from this red circle here, it started around Albany, New York in 2006, and since then it's spread over much of the eastern portion of the United States. Interestingly, there was a case found in Washington State in a cave. Scientists think either one of two things happened. Either a bat migrated all the way to Washington State, or the more likely is that a caver brought it on their gear, either their boots or something, to that cave. So why do we care about this? What's so important about it? There is the possibility of having regional extinction. 9 species in the United States are affected by white nose syndrome, and 3 of those are federally endangered. And at this point, scientists are just trying to manage and conserve the bats that are already in the path of white nose syndrome. And they do provide us with a very important agricultural impact. They eat thousands of insects per night. So they can eat 1,000 insects per hour each night. So they're major pest controllers for our crops. There are some studies being done on treating white nose syndrome. Specifically, last year in Georgia State University, they developed this bacterium called Rhodococcus rhodochrus. And they originally developed it for the delaying of ripening fruit, but they found that it also works as a good antifungal agent for treating bats. They treated 75 bats, all of which survived the winter. The only setback to this is that you can't spray it freely into a cave because it'll diffuse, so you practically have to have the bat in your hand and spray each individual bat, which is inefficient. And Bat Conservation International is also working with Western Michigan University and Ball State University to develop another antifungal called chitosan. And this one's especially promising because it cures the bats of their fungus, and it's also wound healing, so it's kind of doing double duty on the bats. So why downtown Erie, PA? First, that's where our college is, so we decided to stay close. Second, it is the shortest distance along the eastern basin of Lake Erie. So bats, when they're headed south, will stop in Erie for a rest stop after their long journey. And when they're headed north from Erie, they'll stock up on all their insects and get ready for the long journey that way. Here's a picture of where we take our bat walks when we're looking for bats. And pretty much we go across 9th Street, down Peach Street, across 6th Street, and then up through the campus. If you have any particular questions about what buildings we pass by or where we go, you can ask us after the presentation, and we'd be happy to answer those questions. When we do find a bat, we mark its location, how many bats there are, what species of bat, what building we found it at, and then if we can, we mark it with non-toxic paint. And that way we can see if the bat chooses to a different roosting site each night or if there's one that it prefers. And we don't handle the bats because the Game Commission has some pretty strict guidelines on handling bats. 1, it would disturb the bat. 2, they might have white nose syndrome and they could potentially already be immunosuppressed, so we don't want to stress them any further that way. And they could also be potential vectors of rabies. So here are some marked bats. And our hypothesis for our research project is that the number of bats in the Erie area would decrease from the previous census due to the presence of white nose syndrome.
Okay, now this pie chart from 2011 basically represents number of bats marked versus unmarked bats in Getty campus. The reason why most majority of bats remain unmarked is because they were too high on the buildings of the walls, so we can't really reach them even with the ladders. So we just kept them unmarked. In 2012, again, the most majority remain unmarked. 2013, as you can pay attention to the numbers, we have drastic decrease in bat population. 2014, only 7 bats again in campus. We're basically going from thousands to 5 to 10s. 2015, only 13 remain unmarked and 11 marked. 2016, as you can see, that was the worst year, only 5 bats again in campus. 2017, got lucky enough to, there's a little increase in numbers. And this pie chart right here from 2011, going back to 2011, basically talks distribution of bat species again in campus. As you can see from the pie chart, the most majority of the bats were little brown bats, and the remaining is eastern pipistrelle and big brown bats. 2011, again, the most majority is little brown bats. 2013, 2014, going back to 7 bats again in campus. 2015, as we can see, the big brown bats, it seems like they're the most majority of the population. 2016, again, worst year, only 5 bats again in campus. 2017, we're lucky enough to see 1 little red bat. This graph right here basically talks about, so if you look at these 2 peaks right here, in July and August, these 2 peaks represent when maternal pups coming out of roosting, they're learning how to catch insects and learning how to fly. And as we go through August, September, you can see an increase. This peak right here in mid-September basically means they are trying to eat as much insects they can, so because they're going back to migration or in hibernation. 2012, again, these 2 peaks, learning how to fly and catch insects in July, and there's a downslope going back to migration and hibernation. 2013, as you can see, numbers are decreasing, so we're not really getting a good picture here. So only August peaks, again September, and downslope back to migration. And of course, there's 2 peaks, learning how to catch insects and flying. 2014, there's not all the numbers. So there's not statistically significant to even put a picture up here. So as you can see in September, only 3 bats found, but then again, you can see our downslope back to migration and hibernation. 2015, again, there's 2 peaks. 2016, as you can see, we only got 1 bat for every other month. 2017, again. Oh, sorry, go back. Okay, so number of bats per building, again, in campus, as you can see from this table, we got only 4 successful buildings again in campus. The very first one is the east side of Communication Arts Building, east side of Palumbo, and the old main, we'll talk about why is the most successful out of all the buildings again in campus. This is the north side, and this is the east side. This building right here is our east side of the Communication Arts Building. If you pay attention right here, this part of the wall is recessed into the building, so there's less sunshine, less wind, not a lot of people walk by, there's not a lot of disturbance. And what they do is best, they roost right here in this corner. If you walk by, you can see it. East side of Palumbo, again, this wall right here is recessed into the building, not a lot of wind, sunshine, people don't walk by there, so this is a perfect place for them to roost. Old Main, the most popular spot for roosting for various reasons. Number one, if you pay attention to the bricks on the wall, they're kind of bumpy, so they have more surface area to grasp. Number two, it kind of looks like a gray cave. If you look, these windows right here, they have grades on, so that's how bats can tell if it's a cave or not. As you can see, there's 2 little bats right here roosting. Now we'll come back to the east side of the Old Main. If you go down the stairs right here, turn right, you're gonna face this wall right here. On this, if you look closely, there's little bumpy spots on the ceiling. Again, more surface area for the bats to grasp. But the most important reason why this is the most popular spot is because there's a little water right here. The build-up water, as you guys know, insects lay eggs in water, the larvae, and bats know that. So it's a perfect place for them to get the food source and the roosting in this corner right here, the most over this corner and this corner. For the conclusion, as you can see, from 2011 to 2017, we're going basically from thousands to 7, 5, 24. As Maria talked about, in New York, when they got hit by white nose syndrome, it's 2006. According to our data, we can see that we got hit by white nose syndrome between 2012 and 2013. So the question rising from this research, will the little brown bat and the eastern pipit shrill be moved to the state endangered species list, and will they be successful? For our future plans, we're obviously going to continue Project 2018, just because to educate the community, educate getting students, and we're also planning on building bat houses, getting campus just to see if they will stay and hibernate in Erie. We want to thank Tom Reed Center for allowing us to do this presentation. We also want to thank Dr. Stephen Ropsky for advising us in this research. Also, a huge thank to Teresa Catton and Karch Holland for walking with us for our bat walks. These are our references, and we'll be more than happy to answer anybody's question.
Questions for Maria and Ahmed?
You could describe a little bit more about the marking procedure for those pink marks, and I'm wondering in particular if they show any reaction to that when you're marking.
Yep. So let me go back to the picture. These are kind of, it might take a second. So pretty much when we find a bat, we can go back and grab the ladder because we don't always carry it with us. But we'll put the ladder up, go to the bat, and then we mark it. These ones aren't really a good description of what we do, but we go from their neck down their back, and we do kind of a line. And normally we want to see if they're alive. So they'll move a little bit, but they don't fly. They normally don't fly away. Sometimes they have, though. So I don't think it's not toxic. Whether it's disturbing them, I don't think so.
What's the purpose of marking? Are you just trying to track if you're seeing that same bat again?
Yep, so we're trying to see if it's the same bat each day. We have different colors, so we can, if we have multiple at the same time, we can mark them with the different colors. Also, sometimes they prefer like a different roosting site on the same building, or we can find them on a different building a different day, so just seeing where they're roosting.
Are all those species that you've seen over the years, are they all cave hibernating? And seems like it's kind of the big brown bat is maybe less affected. Is that a trend that's happened across the range, or is that?
Do you want to talk about this?
I think you're talking about this right here. Let me go to the slide real quick.
So we find a lot of big brown bats and a lot of little brown bats. Little brown bats definitely are hibernators. Big brown bats, do they hibernate in caves?
Not as much as the little browns do, but
yeah.
So it's the little browns that are being affected because they're more cave hibernators than the big browns.
So according to the data, you can see that we're going from most majority being the little brown, and then we're going back to most majority being the big brown because they spend less time in the cave, so they're less exposed to the white nose syndrome. So they have a higher surviving rate than the little brown bats.
And the little browns are so more used to being with humans. If you ever get a bat in your house, it's always little browns. Yeah.
So if you're finding these little brown bats, even though their numbers have diminished, then they don't have white nose syndrome, so that's a good thing?
So I would think it's a good thing. Whether they are somehow resistant to the fungus, I'm not sure. We could potentially look at that later. But I think it's also just the fact that we don't have 100% mortality. So it's still very detrimental, and we've seen these drastic drops, but we still do have a few. So I just think it's not like completely there's not complete mortality, you know what I mean? Yeah.
Have you seen any exhibiting the white nose syndrome, or have you seen any mortalities like down on the ground, or have you seen any?
No, we have not seen any bats exhibiting white nose syndrome or any with the fungus on them. No. Yeah.
What time of day do you do these walks? Do you do them the same time each day?
So like it depends on the students. Like whenever you have a free time or gap in between the classes. Mine was usually Wednesdays and Fridays around, so I do it before I go home. So it was 11 o'clock, 12 o'clock, like midday.
Yeah. So it's normally during the school day. It's not at the same time. It's different for every student that's going to.
And you don't wanna do this during the nighttime or little gets dark because they're hard to find. They're hard to see.
It is every single day though. Yes. Yeah.
So sometimes you would see, if you were out at night, you would see them flying around?
I haven't personally seen any flying around in Erie. We did recently go to a house in, where was that?
Just south of the casino.
Yeah, south of the casino. We saw them roosting and flying around. And I see them all the time at my house in McCain, but I haven't seen any flying around in downtown Erie. No. Have you?
No.
Thank you.
Thank you.
Our next speaker is Ryan Zimmerman, and he's going to be talking about repeated prenatal VTA exposure results in marked deficits in development of sensory systems in the superior olivary complex.
Is this the pointer in the middle, forward and back? Can I see on the other side?
Sure, if you hold my hand. When you were taping it, live streaming.
Okay. I can just get closer, it's fine.
Oh, okay. I didn't get you loaded up yet. Okay. There's a mouse there.
So good afternoon, good late morning. I'd like to say that was a very interesting talk about the bats. I also like how you coordinated your shirts. One of the first things I noticed. So my name is Ryan Zimmerman, and I'm a first-year medical student at the Lake Erie College of Osteopathic Medicine. And this research was conducted in partial fulfillment of my master's degree in the Auditory Research Center at LECOM in the lab of Dr. Randy Kaleza. And so thank you again for the introduction, and to Tom Ridge Environmental Center for allowing me to present today. I'll be discussing that repeated prenatal VPA exposure results in marked deficits in development of sensory systems and a superior olivary complex in a rodent model of autism. And so autism spectrum disorder is defined by the DSM as a persistent deficit in social communication interaction, as well as a restricted, repetitive patterns of behavior, interests, or activities. What's interesting about this spectrum from least severe to most severely affected individuals is that a vast majority of them have an auditory dysfunction. Now, this auditory dysfunction can range from complete deafness to hyperacusis, in which normal sounds are perceived significantly louder by the individual. Now, before we get into the specifics of how we observe the auditory dysfunction, I believe it's relevant to discuss some anatomy. Now, big picture, how you hear sound comes in as a sound wave and transverses the outer, the middle, into the inner ear, where the cochlea then converts the wavelength of the sound into a neural impulse, which is then projected to the cochlea, from the cochlea to the cochlear nucleus through the vestibular cochlear nerve. And so the cochlear nucleus is what is receiving the input from the cochlea. Now, it's easiest to imagine, I'll say it on this side, imagine this as your left ear. And so if I play a sound in your left ear, your left cochlear nucleus makes a major projection to the superior olive by way of the medial nucleus of the trapezoid body. Now, this region is really important for your ability to localize sounds, localize whether my voice is on your midline or away from the midline, as well as to listen in background noise, as to filter out background noise. So the MNTB receives input from the cochlear nucleus contralaterally, and then this MNTB is terribly interesting in that it's a sign reversing relay center, in that it receives excitation, it receives excitatory glutaminergic input, and then it projects negative inhibitory input to the corresponding regions, the superior paraolivary nucleus, the medial superior olive, as well as the lateral superior olive. And so when you hear a sound in your left ear, it's important for your right SOC to have a dampening effect and to enable you to understand that it's coming from the left side. Now, you see here with these regions that are marked high and low, all of these regions are tonotopically arranged, meaning that cells fire according to the sound, the frequency of the sound that's being perceived. Even right now, whether my voice is low or the pitch gets higher, those cells only fire accordingly. Now, this is messed up in individuals who are experiencing autism. Now, one way that we can look at this is through a drug called valproic acid. Now, historically, valproic acid has been used in the treatment of epilepsy, bipolar disorder, as well as migraines. And so women who are taking valproic acid for epilepsy, their children, their offspring, are at a 20 times greater incidence of experiencing the characteristics of autism spectrum disorder. And so when an individual has been prenatally exposed, they have drastic changes in neurons of the superior olive that include size, number, shape, and morphology of the cells, as well as abnormal tonotopic maps. So what we expect then is abnormal ability to listen to background noise and the inability to localize sound sources. So two things that we were interested in within studying the SOC are calcium binding proteins. Calcium binding proteins are absolutely essential for your cells to fire normally in order to respond to different stimuli. And so two that we were interested in are calbinin and calretinin. Calbinin is expressed by cells of the cerebellum and the MNTB, and it's actually expressed by the cell itself in the cell body, whereas calretinin is expressed, as I discussed, the projection from the cochlear nucleus to the contralateral SOC. It's expressed by those globular bushy cells. And so what we know is that they're absolutely required, and it has been shown that in humans with autism, these are reduced. And so how exactly do we go about exposing a rat to valproic acid? So we do time mating, and we check the cage for the presence of a vaginal plug, which denotes embryonic day 0. And then we actually use peanut butter to give as a vehicle for the drug. And so on days, you can see here with our little timeline here, on days 7, embryonic days 7, 8, 9, 10, 11, and 12, we give the rat 3.1 grams of peanut butter. In the experimental group, on days 10 and 12, we give 800 milligrams per kilogram of valproic acid mixed in 3.1 grams of peanut butter. And we do this for two reasons. One is that peanut butter and oral ingestion most replicates the mother who is orally taking valproic acid. And we choose days 10 and 12 because that's the day that neurons of the superior olive are born in the rat. And so after the exposure, the normal protocol is that the rats are kept with the mother until 3 weeks. We wean them off, and then at 4 weeks, they're anesthetized with pentobarbital, and then we do a surgical perfusion, remove their head, take their brain out, and then slice down the brain, and then we stain it using primary and secondary antibodies to stain for calretinin and calbinin, as I previously mentioned. So the first aim of the project was to really look at gross sensory development by way of looking at the ears and the eye opening. And so what we noticed was that there is a significant delay in valproic acid exposed compared to control. And so the chart, the graph that we have here is looking first at the ears opening, and what we did was at 50%, they had to have both ears open in the sense that you could actually hold open the pinna and observe down into their auditory canal. And what we noticed was at 50% and at 50%, there was a significant difference by chi-square between control and VPA, as well as when there was 100% of the controls open. And so it was significant that the ears of the VPA rats, they were closed. They were actually, sealed over. And so the same thing was observed with the eyes. And the eyes is a little more easy to observe because you're looking directly at them. And we noticed the same thing. So we ran the same statistical test in that looking at 50% at days 15 and then at 100% the next day, 100% of the controls had all their eyes open. It was about a 3-day delay for both, and that when all controls had all their eyes open, there was actually still a cartilage, like a seal over the eyes of the rats that were exposed to BPA. And so we know that the gross development is wrong. We know it's delayed. And so that led us then to question, how are these rodents, how are these rats then expressing calcium binding proteins differently? And so the next step was to look at calbindin expression. Now, A and B on the top portion is actually looking at octopus cells in the cochlear nucleus. I wish I had time because it's really interesting to get into. But we'll just be looking at here in C. This is from a control rat. And what you'll notice is that in the cell body, it's positively expressing calbindin. A vast majority, most of the cells are expressing calbindin positively. The yellow triangles are calbindin positive, and then the red are calbindin negative. So most of them really lights up green. And then if you look at VPA exposed in D from a similar sample, same age rat, there's a significant decrease in the amount of calbindin expression. And so you can see same color coding is that red is no expression, yellow is the positive expression. And one thing that we found interesting about the VPA exposed was that here in white, there actually is calbindin expression, but it's localized subcellularly and restricted just to the nucleus. So there's some sort of trafficking issue, whether it's processing from the Golgi, that it's not actually getting out into the cell body. Just something that we observed and found interesting. And so this corresponds to the graph down here at F. And what we noticed is that by Mann-Whitney U, non-parametric Mann-Whitney U, there was a significant decrease in the percentage of cells that were expressing calbindin positively. So then the next name was calretinin. Calretinin is not expressed by the cells, by the principal cells themselves. It's expressed by the cochlear nucleus cells in their projection across the midline and in the calyx-like terminal that terminates on those cells, the MNTB, that I mentioned it's very important for your ability to localize sound. And so you can see here, everywhere there's green, that's a calyx ending on those MNTB cells. So the cells themselves aren't positively expressing it, but you'll notice in the white that there are a few cells that don't have that. Most of them do have it. There's one here that doesn't. But then if you look over here at the valproic acid exposed, there's not much expression of calretinin going on. And so what this enabled us also to see, going back over here, is that you can view the axons as they actually come in and terminate on the cell. And so that indirectly enabled us to measure the axon diameter. And so looking at close to 2,000 axons, we noticed that there was a significant decrease in the size and the caliber of the axons that VPA exposed compared to the control. So now we know the sensor is delayed. We know the eyes and ears are opening more slowly. We know that the calbindin expression is decreased, the calretinin expression is decreased, and the axons of these cells are also decreased. And the significance of that is just like a telephone wire that runs to your house. The larger the diameter of the telephone wire, the more efficiently that the signal is going to reach. Comparatively, with a smaller signal, it's easier to get lost and doesn't transmit as nearly effectively. So that led us to our final aim, was to look at these actual calyces, these actual terminal ends of the MNTB. And so this is really cool. What we did was, this is actually a gross brain. So this is a fresh brain, hasn't been preserved. The same day, it's a 28-day rat. And what you're looking at here is, this is the injection site. This is the trapezoid body, where those axons are actually crossing the midline. We put in a lipophilic deposit tracer called Di-I, and it crosses myelinated axons, and then interrograde labels the regions that they bind to. And so here you see the injection site in B, where the tracer went into, and then you can actually see the axons as they go out to the regions of the SOC. So then we use a immunofluorescent scope to zoom in on calyces themselves. And that's what you're seeing here in C. You're actually seeing calyx wrappings on actual terminal endings on the cells themselves. And so what we did was, this is like 100 micron thickness tissue, and that's like about half the width of a wet tissue paper. It's extremely small. And so what we do is we actually took it to Penn State Barron, where they have a confocal immunofluorescent microscope. And what it enables us to do is take that 100 micron sample and cut it way down to like whatever the distance is of the actual calyx itself, say 5 microns. Then it takes stepwise scans and enables us to get a stack, which we can 3D reconstruct into what the axon itself looks like. And so here in D, you're looking at a control reconstruction of the actual calyx terminal that ended on the principal cell of the MNCB. And so here's another one. This is a VPA exposed, and another VPA exposed. So really what this enabled us to do was look at the volume, the surface area, the ratio of the two, and then the ratio of the calyx surface area to the surface area of the MNCB. Now, we didn't observe any differences in terms of the surface area or the volume, or the ratio, but what we did observe was a significant difference in the rats exposed to VPA. They actually had a greater calyx surface area and relative to the MNTB surface area, which should seem counterintuitive. We have smaller cells, we have fewer cells, expressing less calcium binding proteins. And now we're seeing that there's a greater expression of these inputs. And so it kind of fits in with the hyperworld theory of autism, in which there is actually an overabundance of excitatory inputs, which may lead to those repetitive behaviors whenever they're in very loud sounds or otherwise unfamiliar environments. And so that led us on to future research, which is looking at projections higher in the chain to what is known as the inferior colliculus. And so in conclusion, actually I have hopefully this will be able to play here. The total calyx enabled us, if we make a 3D stack of it, it actually enables us to make a video, which hopefully will. So that's actually the axon that you're seeing here. This is the axon, and then it actually terminates out on the principal neuron of the MNTB. Science is so cool. And so what we notice is that these rats that have been exposed to VPA, there is a greater surface area in spite of the decreased axon diameter, decreased calcium binding expression, and delayed sensory development. So in conclusion, VPA results in a delayed sensory development and in both the auditory and the visual system, and we observe decreased calbindin, calretin expression, as well as axon diameter. Now, what this would result in rats is a little bit difficult because they are in a lab, but in the environment, it would result in their inability or difficult listening and background noise, which could be very dangerous for an animal in the environment. And then taken back to humans, a difficulty listening to and interpreting speech and those cycles of repetitive and restrictive behaviors. Thank you.
Questions for Ryan?
So if this is happening like before they're born, they're still in the womb and you're taking them out and seeing this lapse of development, right?
So this is after they're born. So like, yeah, they're exposed prenatally. They're exposed on those days 10 and 12.
Oh, okay.
And then their mother gives birth. They're kept together for 3 weeks, and then on the 4th week, that's whenever we perfuse them.
So do you predict that one that was exposed versus one that was not exposed, would the one that is exposed ever catch up, like after it was 10 weeks, 12 weeks, by the time that they're both at 12 or 15 weeks, they would be equivalent, or there's always going to be that delay?
Sure, that's a great question. It really is a great question. And so the question is kind of like a compensation mechanism, if you will. Do those rats that have been exposed prenatally, are they able to regain what they've lost? I think it would be an interesting study, although there would be a delay. I think if you looked at 15 weeks, if you looked at 18 weeks and 20, you would still see, because basically how it works is the architecture that you have is what you have. The number of cells that you have today, we should be grateful for them because it's going to be less tomorrow. And that's kind of the same thing with those control versus the VPA exposed. And so if they have less neurons and the neurons themselves are just misshapen, it's going to be difficult for them to ever get back to what a baseline would be. Now, there are studies that they've done, training and in which, experience is such a wonderful thing. And so you could, now I guess this would be more relatable to a human, if you were to, especially why early intervention is more important. The earlier that you intervene, the more likely that that individual is going to have less of a deficit.
That makes sense. Thank you.
Other questions?
It's valproic acid. Is that anything? Is there any natural exposure to that, or is that only via this epilepsy drug? Is there any environmental exposure to that drug?
That's a good question. It is a synthesized compound, and so it's also known in pharmaceuticals as Depakote is another name for it. Now, there is some environmental exposure that relates to. So, like, if a majority of the cases of autism are multifactorial, genetic predisposition, environmental exposure, this is one known isolated cause that, replicates similar between rodents and humans. Do you have a question?
Yeah, so is there a take-home message about using valproic acid as a treatment, and that kind of stuff? Or?
Well, I think the uses of it has waned off in recent years. Now, how it works is the research itself indicates that it kind of regulates genetics through epigenetics. It doesn't seem as though it's an incorporation into the genome itself. And so if a woman was to take valproic acid for a course and then cease taking it and then become pregnant, it doesn't seem as though there's a significant risk for autism. But there's something in that ability that while it's taking, that it's able to cross and cause damage to those neurons.
When was this discovered, this connection between this valproic acid and autism? I didn't know that there was, causes for autism that could be identified.
Yeah, it really, so the original paper, her name was Patricia Rodier, and she actually was one of the first people to describe the link between, basically how she first described it was as a superior olivary complex knockout. And so when she was exposing these rodents to it and she was looking at the cells, it was like, there's a significant link here. She was also one of the pioneers in the link between autism and craniofacial abnormalities. And so this is probably in the mid-90s that the landmark paper came out for it.
Okay, thank you.
Thank you.
Jeanette. It's my pleasure to introduce Jeanette Schnars.
So that concludes the session. If you were a presenter today, you should have a lunch ticket. If you don't, go see the registration table. You're welcome to stay for lunch. And we will do the student awards at 1 o'clock in this room. And thank you, everybody, for participating. It's a great session. Thank you.